Hi everyone, just reaching out, hoping all you guys are doing well. I can't believe that we have been out of the classroom this long. I miss being with you guys every day in the classroom. Wish things were a little bit different, but I kind of learned how to put myself on the video. So I'm going to try it out this time, see how it works, but I wanted to reach out to you guys. Let you know that I'm thinking about you guys every day, and that I hope you're doing well. And if you guys need anything, let me know. Send me a quick message if there's something going on, and you need a little bit more time to get the work in. Please, please, please send me a message. Let me know what's going on. I have the best interest for you guys. I want you guys to do well. Later this week, I'm going to try and send out about the grading policy and how important it is that you guys keep up on your work. But if there is something at home, number one priority is your family. I want you to put them first, but please reach out to me. But fully understand that I truly miss you guys. I miss MacArthur. I miss being with you every day. But we're going to start our next video. It's going to be on protein synthesis. And it's a little bit more difficult, but I'm going to try my best to explain it to you guys. And if you guys have any questions, just reach out to me with questions, and I'll attach other videos. But it is a little more difficult, but I'm going to try and break it down as easy as possible. So in some of these slides, I'm going to play so that you guys can see the animation. Some of them I will not play. But let's first start on our first slide. So protein synthesis. So we've learned about DNA, we've learned about RNA, we've learned about DNA replication, and now we're moving on to protein synthesis. But you guys already know we started at the beginning of the year with the word synthesis. And the word synthesis means to make or build. So we put the word protein in front of it. We are making proteins, and we're making or building proteins. So we're going to learn how we take our DNA and we build it into a protein. So this video might be a little bit longer than normal because it does involve two major concepts called transcription and translation. But the overall concept is to make new proteins, and that originally comes from your DNA. So we're making proteins all the time. We know based upon biochemistry that proteins are made up of 20 amino acids. These are essential amino acids. We learned about amino acids and their building blocks for proteins. So I need to build a polypeptide chain in order to build my new protein. And there are 20 essential amino acids. And, if we were in class, I would ask you, how would you get those amino acids? And you guys would tell me you would eat them. And then our body just takes them and puts them back together. And we, always have that blue protein in front of my room, and I show it to you guys all the time, and I see each one of these marks as an individual protein. I wish I had that model in front of me today, but it's in the classroom. So we have the sequence of amino acids, and, remember our blue model had these black lines on it, and every black line represented an amino acid. And you guys already know that blue model was one big long strand, and it had to bend and fold in order for it to give it structure and function, the two words that I use all the time. So proteins are really, really, really important. They're a really big deal. They're your enzymes, your hormones, the most really important hormone, insulin. They are antibodies, and everybody's talking about antibodies today. And these, your body has to, go through protein synthesis to make new antibodies, because we always want to have these antibodies for the rest of our lives. And each protein has a specific structure. That specific structure leads to a specific function for the protein. And, these proteins are essential for you to live. And these proteins are made up of amino acids. And they specifically, amino acids come from your genetic code, which is your DNA. Okay. So this slide just tells us cells store vast amounts of coded information in their genes. And we learned prior in the other PowerPoint that I told you your genes are located on your chromosomes, and then inside your genes are your DNA, which is the smallest unit of genetic information. And the ribosomes construct your protein. So we're going to kind of learn that later when I get into this constructing the proteins as translation. So DNA protein connection that I'm just talking about here, that you kind of, think about it, and I would ask you in class, like, who do you look like? And most of you guys are going to tell me you look like your mom, or you look like your dad, you look like your brother, you look like your sister, you look like your grandmother. The reason why we look like each other is because we have similar proteins. Not the same, but we have similar proteins. Like, I look like my mom. Because I make similar proteins that she has, and my Kaylin and my James look like me because they make similar proteins from my genetics, and my Liam, he looks like my husband because he has similar traits or proteins as my husband. So protein-dependent traits can be the type of hair that you have, your eye color, certain diseases. So the first thing we're going to start off with is transcription. Okay, so there's two major steps here. There's transcription and there's translation. So how I remember transcription comes first. It has the C, and translation comes second. It has the L. So we're going to start out with transcription, and transcription is a process that takes place in the nucleus. And the first thing in order to start transcription is to find the DNA strand or the DNA pattern that we want to copy. And we already know that who can copy DNA's code is messenger RNA. Messenger RNA can take DNA and convert it to its code. So DNA nitrogenous bases are ATGC, but in RNA they AUGC, which we already know. And the DNA code gets transferred into the mRNA code. And the reason why DNA has to turn into mRNA is because DNA is double-stranded, as we know, and RNA is single-stranded. So mRNA is easier and can leave the nucleus, where DNA cannot because it is double-stranded. So the message is being sent out by mRNA. And there's a lot more that happens that I'm not telling you about now, but when you take AP Bio, we can tell you about that later, where there's good DNA and then there's junk DNA, and we want to cut out the junk DNA and put together the good DNA. Not something for you guys to worry about now, but they are called introns, then exons, and we want to put together the coding DNA, which are the exons. Anyway, okay, so that is transcription. The nucleus, mRNA comes in and copies DNA's code. And now here is just a picture showing our starting point, where we have our double-stranded DNA molecule here in blue, and it's going through transcription, and it's turning this pink color here because that's mRNA, and the base letters are changing, and you notice that it's single-stranded. This does have to go through further processing, and it will get shorter, it will get a head, it will get a tail. The head tells it where to go, the tail tells it how to get there. But in order to do all this, there's a whole bunch of enzymes involved, and I kind of mentioned them last PowerPoint, where there's helicase that has to come in, and there's another one called RNA polymerase now. RNA polymerase helps to build the new RNA molecules. When I talked about it prior with DNA replication, I was talking about DNA polymerase, but you notice I'm saying ACE, helicase, polymerase. They are enzymes, and they're going to match up. So the U and A and the G and the C. So this is transcription, and we're trying to make our new RNA sequence that can leave. And you notice the little pores here. That's how the mRNA can leave. And the mRNA's head is telling it to go to a certain ribosome. And at that ribosome will then go through translation and try to make its new protein. So here, I think I'm just going to play this slide. Bear with me. Is showing, hold on. Oh, it doesn't play that much for me. Okay, so anyway, so you are looking at your DNA strand down here. How do I know that that's DNA? How I know that that's DNA is that there are T's. And now you notice that here comes my new strand. Remember I was talking about the semi-conservative model where there's an old strand and there's a new strand. You notice the blue and then you notice the black. And then you notice all these new nitrogenous bases coming in to match up with its corresponding DNA here. So this is RNA being built. And how do I know that these are RNA? You notice that U. And how do I know this is DNA? I notice the T. And so the RNA bases match to the DNA bases for that strand there. So, all right. So now we have gone through transcription. So let's go start translation. Translation, again, is your messenger molecule going from the DNA to the ribosomes, moving from the nucleus to the cytoplasm. That's really important to know. Transcription is in the nucleus, and translation is in the cytoplasm and comes together at the ribosomes. So your mRNA is now going to match up with something called tRNA. tRNA is the transfer RNA, which you already know because we went through the 3 types of RNA already with your reading comprehension. And tRNA matches up with mRNA and brings in the proper anticodons. So mRNA is the codon and tRNA is the anticodon. And the codon, mRNA, is the most important because it's carrying the code of life, which is DNA. So mRNA is the codon, tRNA is the anticodon, and tRNA free float in the cytoplasm, and they match up just like puzzle pieces. So mRNA and tRNA, they match up. I'm going to tell you guys in a little bit on how you actually figure out what amino acid does match up. And then from there, you're building your new protein chain. And the last type of RNA, which is not on here, is called rRNA, which is ribosomal RNA, where you're building your new amino acid after amino acid plus amino acid, giving your polypeptide chain. And that polypeptide chain is going to bend and fold, giving structure and function to express my new protein. But that protein's also going to have a tag on it to kind of tell it where to go. You know, maybe it's a new cell membrane, maybe it's a new antibody. Okay. So here is my new strand. I see that here. So I have my DNA. Now it's converted to mRNA. You notice my new strand here. And eventually it will be read across. The strand will be read across. So then you could build your new amino acids. So it starts out with DNA, converts to mRNA. You have your strand here being read at the ribosome. tRNA will be in the cytoplasm, bringing in the new amino acid. And let's see if we have any graphics here. So here the ribosome just comes along and reads the letters. And I'm going to tell you it's going to be read 3 letters at a time. And it's going to bring in the new tRNA so that we can get our growing amino acid chain, because that's the whole point. We're trying to build this polypeptide here, but to get this polypeptide to work, I've got to get it to bend and fold to its primary, secondary, and then tertiary shape. And then it gets tagged and it tells it where to go. Okay. Now looking here, this is a chart that I would bring up all the time, and if we were looking at it in class, but we're not in class. So you have your 3 letters. So I want you guys to look at it this way. Here's your first letter. Here's your second letter. And then here over here is your third letter. So here's your first letter, my second letter, and then my third letter. So if I wanted to start out with the one that every single protein starts with, that is going to be A. Here's my first letter. So I'm going to stay in here. U is my second letter. And then over here is my G. So the easiest way for me to read this is I look at first letter, second letter, third letter. But you could just look here if you had to. So you're going to go first letter, second letter, third letter, or if I said AUG, you could find AUG on here. But I started with that one because every single protein starts with AUG. Now, if I asked you another one, like U. So my first letter is U, my second letter is U, my third letter is U. So if I look here, U, and I go across U, it is PHE phenylalanine. Now we abbreviate these with 3 letters. It's not called PHE or LEU. They're abbreviations, which we will use. So this is leucine, this is phenylalanine, this is serine, this is proline, valine. Not necessary for you guys to memorize them at all. You will be given this chart, and you look at, again, first letter, second letter, third letter. So if I said to you guys G, U, C. So I'm going to give you a second. I'll give you a second. Look, G, U, C. Okay, I think that's more than a second. Anyway, so G, U, and then I come down here to C, which is going to be valine. That's how you guys are going to tell me what the mRNA is. It's 3 letters, giving your amino acid, and again, you get these amino acids from what you eat, and then they are put back together by protein synthesis. And also that there are stop codons. So UGA, UAA, and UAG, which I believe come up here, are your stop codons, and they have to stop the sequence because the polypeptide chain just can't keep going on and on. It has to stop, so it has a start, it has a stop, then it will bend, folds, and then go to its proper destination, whatever that protein may be, enzyme, hormone, antibody. Proteins are so important. We're sitting here right now, we're all making proteins. So I'm going to just go off of here, see where I am. So now at this point in translation, I am leaving the nucleus. I have my mRNA strand. I have my strand that I want to be coded. And then the tRNA comes in, brings in the proper amino acid. And that amino acids come from the codon chart that I just showed to you. So amino acid, amino acid. So here's my growing polypeptide chain, which is growing out of the ribosome. Okay. And then here shows your DNA matching with mRNA. tRNA comes in free floating from the cytoplasm, and it matches up. So you've got your codon, I've got my anticodon, and I start with MET. Just like I told you guys, every single protein should start with MET. Then from there, it builds your new polypeptide. On here I did have codon and anticodon. So this is building your new polypeptide chain. Here again showing your DNA to mRNA. So DNA to mRNA again is transcription, and mRNA to your new protein is translation. And then you build your polypeptide. It's made up of, there's only 20 amino acids. So if I went back to that chart, you could see that there were 4 of them that coded for valine. So, and I'm going to talk about it later. So if there was a mutation of valine and it switched one of the letters, it might be okay. Might not. But for valine, it's kind of good because there were 4 of them that coded for valine. But the ribosome is really important. That's what's building our new protein. And, we've talked about protein synthesis, all year basically, but you guys really didn't know what it was. So here just showing you, my blueprint, my DNA is in the nucleus. It has to get converted by mRNA. Then I have my ribosome factory, which is my ribosomes to build my new proteins, and here's my polypeptide chain, and then it has to bend and fold. And here again, showing you DNA transcription. Your mRNA is translation. You get named protein, and look, here's my protein. I've shown this to you guys before. This is kind of, think of this as my blue model that I have in the room all the time. And then it makes your traits, your hormones, your antibodies, your enzymes. Proteins are so important. Protein synthesis is so important, and we're doing it all right now. So you got transcription, you've got translation. And again, I'm showing you, again, let me try and play this one. So you got my DNA, okay, to my mRNA, which we already know, which is transcription, right? And then we have here, so mRNA is my new protein, that's called translation, I think you guys are telling me. And then I have my ribosome coming along. It's going to read how many letters at a time. And hopefully you guys should be saying 3 to yourself. So it's going to read 3 letters at a time. And tRNA is going to come in and bring in the anticodon. And then I should have my new protein chain coming off my code. Okay. Oh, notice here, nucleus, cytoplasm. Okay. And then my tRNA is in the cytoplasm, bringing in the new amino acid. Okay, so over and over it will be done. And then here is just a big overall picture of everything kind of that's going on. You got your DNA. DNA has to open up. Remember, it has to open up, and you have helicase breaking those hydrogen bonds to open it up. Wish I had my DNA model. Anyway, and then your one side of your DNA is going to get read, and now I'm building RNA, so it's called RNA polymerase. So you notice all these enzymes, helicase, polymerase, these are proteins. So protein synthesis is really important, making new proteins. So you notice my mRNA code sneaking out here through the pore, and then it's this mRNA is coming to the ribosome. Notice the ribosome is made up of 2 pieces. And then notice here, you've got your tRNA free-floating in the cytoplasm. tRNA has to go find its correct amino acid to match up to the corresponding mRNA 3 letters. And they match up because they come together like pieces of a puzzle. And you notice here, my abbreviations for my 3 amino acids. I've got leucine, proline, aline, phenylalanine, glutamine. My polypeptide chain is growing. And I've got my 2 major processes here called transcription and translation. This is a hard topic. You might have to watch this again. Message me if you guys have any questions. But remember that I'm thinking about you. Wish we were back in the classroom. You guys are doing an amazing job. Please keep up the good work. And I'm going to come back with a video about the new grading rubric and how important it is for you guys to obtain the 4. I hope you guys have a great day. Message me if you have any questions. Bye.